Dear students, let's continue with the cell cycle. In the previous lecture, we briefed about the cell cycle and what are the factors that affect the cell cycle. Now let's continue with that. You know that the cell cycle involve all the events and measures which uh, lead to the division of the cell. These include all the cellular events, be it the growth, be it the taking the nutrition, and then proliferation, and then finally division. Uh, is called the cell cycle. We have discussed it in the previous lecture. So now today we will discuss uh, the phases of the cell cycle. Cell cycle has two main phases. There are two main phases of cell cycle. Cell cycle has two phases uh, through which the cell progresses, uh, grows, divides into two daughter cells, and again repeats this. So this is there are two phases. Let's discuss these phases. The first phase of the cell cycle, the major two phases, the first phase of it, that's interphase. Interphase. And the second phase is the M phase. So these are the two important major phases of the cell cycle. We're going to discuss about these one by one. So what is interphase and what is M phase? Let's start with the interphase. Let's start with the interphase. Interphase. Uh, Previously, it was thought to be the resting stage or resting phase of the cell cycle, but it is not so. This phase is very active phase, metabolically active phase. It's not a resting phase, it is active phase, but the changes which are taking place in this phase are not visible to the eye. That's why it was thought to be a resting phase. But there are very changes, very important changes taking place in this phase. Metabolically active phase is this phase. So this is not a resting phase, which it was thought to be previously thought to be a resting phase. But there are very important changes which are taking place during this phase. This is interphase. Now, what is interphase? This interphase actually uh, is also called the preparatory phase of the cell cycle. It is actually the preparatory phase. This phase involves the preparation for the next phase. That's the M phase. That's the division of the cell. So in this phase, in interphase, the cell prepares itself for the division. So this interphase is also called the preparatory phase. Preparatory phase. So this is very important phase as the cell prepares itself for division, for dividing into two daughter cells. So this is preparatory phase. This interphase is now divided into subphases. First phase is G1 phase, second is S phase, third is G2 phase, G2 phase. So this interphase is divided into three subphases. We have seen here it's divided into three subphases. So this interphase, which is a preparatory phase for the cell, and it is now divided into three subphases. First subphase is the G1 phase, second is the S phase, and third is the G2 phase. So these three phases are the three subphases of interphase. So after these three phases, the completion of these three subphases is followed with M phase. If we show it diagrammatically here, let's show it first diagrammatically. This is the cell cycle. This is G1 phase, this is S phase, this is G2 phase, and this is M phase. So we can say, show here, diagrammatically we can show that this is the G1 phase, this is S phase, this is G2 phase. So G1, S and G2 phase, these three phases from here to here, this is interphase. Interphase. So we can say from here to here, this is the interphase. Interphase. After interphase, we will follow M phase. That's mitosis. We will deal it later. So these three subphases of the interphase, G1 phase, S phase, and G2 phase, follow one after the other. And then after completing the G2 phase, that is the final phase of the interphase, it enters into the M phase. That's now mitotic phase, division phase. This is the second major phase of the cell cycle. Now let's see let us see these phases one by one. What changes are taking place in these phases, one after the other. So let's see these phases. If we see about the interphase, the preparatory phase, let's make it clear. Before the cell has to divide, it has to make all the preparations, all the things available, all the nutrients available, all the organelles available in order to divide into two equal cells. Because, for example, if we have to, if a household has to, there are two people living in a household, they have to separate. Before separation, they have to make arrangements for separating into two new households. They have to make availability of the food stock, the availability of everything they need, and then after they are in a position to separate or to divide into two new households. Likewise, the cell has to make preparations before it enters into M phase or before it divides into two daughter cells. Such whole set of preparations are done in this interphase, which involves these three phases: G1 phase, S phase, and G2 phase. Now let's take these phases one by one. Now let's first is the G1 phase. G1 phase. G1 phase is also called gap 1 phase. Gap 1 phase, or it's also the growth phase. Growth phase 1. That means this is why it is called a gap phase, because it is the time interval, it's the duration of the time from the previous M phase. Previously, the cell which has divided through the M phase division and through the mitosis, it is the time interval, it is the gap from the previous M phase up to the up to the next, we can say up to the from previous M phase to the next phase, S phase, we can say it is a gap. It is the gap between the previous M phase and to the S phase, that synthesis phase. That's where DNA replication takes place. So the gap between from previous division up to this synthesis phase, that is S phase, is called the gap first. Actually, it takes this gap up to first division, a cell when it divides, the next cell which it has to divide again, but it readily will not divide. It will accumulate the nutrition. It will just start to grow in size, and then after growing in size, dividing the organelles, uh, pulling into the all the nutrients, and then after it's able to enter into a next stage. So this stage gap period where it grows in size, takes nutrition, accumulates nutrition, this is called gap. This period of gap between the previous M phase and the synthesis phase. So, or it's also called a growth phase because the cell grows here. In this phase, the cell starts to grow, starts to accumulate the nutrition. That's why it's also called growth phase or gap one phase. Or it's also called pre-synthetic phase. It's also called pre-synthetic phase or pre-S phase. So now, one thing we must clear here: any cell which is coming in this cycle, from starting from this G1 phase. This G1 phase, actually the first phase is a preparation for the next stage to commence. That means कि जो जब ये सेल G1 फेज में आती है तो ग्रोथ करती है, उसके बाद उसको S फेज में आना पड़ता है. एक्चुअली S फेज का होना इससे पहले इस फेज जो इससे आगे आएगा फेज, जैसे S फेज है, the phase which is after that, the preparation for the S phase are all performed in the previous phase, that is G1 phase. इसका मतलब होगा कि S phase में जो है सिंथेसिस फेज, रेप्लिकेशन होगी डीएनए की, इस फेज की प्रेपरेशंस पहले हो जाएंगी इस G1 फेज में. उसके बाद जैसे S फेज में रेप्लिकेशन होगी, उसके बाद जैसे G2 फेज आ गया, G2 फेज के बाद M फेज, तो सेल M फेज में नहीं आ जाएगी, सेल M फेज डिवाइड नहीं करेगी जब तक न सारी तैयारियां पहले ही हो जाएंगी जो M फेज में जरूरत पड़ती है. To make it understandable that if a cell now has enter into S phase, it has to enter into the S phase. Before entering into the S phase, before replicating the DNA, it has to make all the possibilities, all the things available, all the prerequisites which are necessary for replication of DNA. That's a major event taking place in the cell cycle, and especially in the S phase. So all the preparations are to be done. These preparations are done in the previous phase. That means the G1 phase makes all the availabilities, preparations for the S phase to commence. And then likewise, the G2 phase will enter into M phase. It will not enter into M phase. The cell will not enter into M phase unless and until it will not prepare or make all the availabilities which are required for the M phase to perform, which are required for the cell to divide. So कहने का मतलब ये है कि जैसे सेल G1 फेज में आ जाएगी, ये तैयारी करेगी सारी चीज़ों को जो उसको ज़रूरत पड़ेगी S फेज में. इसका मतलब हो गया जो चीज़ें ज़रूरत पड़ेगी S फेज में वो पहले फेज में बन जाएगी G1 फेज. इसी तरह जो चीज़ें M फेज में ज़रूरत पड़ेगी वो G2 फेज में बन जाएगी. तो ये कोऑर्डिनेटेड मैकेनिज्म है, कोऑर्डिनेटेड इवेंट्स होते हैं इसमें. तो पहला फेज जो है वो है G1 फेज, गैप 1 फेज और ग्रोथ फेज 1. तो हम देखेंगे, लेट्स सी नाउ हियर दैट these are the preparations for the S phase to kickstart to commence. What actually are the preparations? You know that जो S phase होता है इसमें we know S phase में होता है synthesis. It is a synthesis of DNA. We can say it is also called synthetic phase. Synthetic phase. एस फेज को कहा जाता है सिंथेटिक फेज, दैट इज देयर इज सिंथेसिस, देयर इज फॉर्मेशन ऑफ और रेप्लिकेशन ऑफ डीएनए, रेप्लिकेशन ऑफ डीएनए। तो एस फेज में डीएनए की रेप्लिकेशन हो जाती है, डीएनए का जो अमाउंट होता है वो डबल हो जाता है। वी नो दैट हमारे पास जो सेल होती है वी हैव 46 we have 23 pairs of chromosomes. This amount of the chromosomes, the amount of the DNA, the number of the chromosomes, the amount of the DNA is double. That means this is the replication of the DNA taking place. For replication to take place, we need certain things. We need certain things. We need certain instruments. We need certain things to make this replication of DNA a possible one. That means कि अगर हमें यहाँ एस फेज में डीएनए की रेप्लिकेशन करानी है तो रेप्लिकेशन कराने के लिए हमें चीज़ें चाहिए पहले। फॉर एग्जांपल जैसे हमें वी हैव टू कंस्ट्रक्ट अ बिल्डिंग और हाउस, फॉर दैट पर्पस वी हैव टू मेक ऑल द अवेलेबिलिटीज, ऑल द प्रीरिक्विजिट्स इन एडवांस। फॉर एग्जांपल अगर हमें एक मकान बनाना है तो हमें उस मकान बनाने के लिए पहली सी चीज़ें तैयार रखनी होती है। For that purpose, we have to get the stones, we have to get the bricks, we have to get the cement, we have to get the water and every sort of things which are available in advance, so that we can construct a building, a house. Likewise, जब हमें एस फेज में डीएनए को रेप्लिकेट कराना है, रेप्लिकेशन कराने के लिए हमें एंजाइम्स की जरूरत पड़ेगी। We need enzymes for replication. We need nucleotides for building this giant 6 feet long DNA molecule, new molecule replication. तो हमें यहाँ पे न्यूक्लियोटाइड्स की जरूरत पड़ेगी, एंजाइम्स की जरूरत पड़ेगी। सो ऑल दीज थिंग्स हमें ये सारी चीजें चाहिए होती है। तो यहाँ पे एस फेज की टाइम हमें वो प्रॉब्लम नहीं होनी चाहिए, हमें ढूंढना ना पड़े कि हमें न्यूक्लियोटाइड्स की अवेलेबिलिटी नहीं है, एंजाइम्स की अवेलेबिलिटी नहीं है, प्रोटीन्स की अवेलेबिलिटी नहीं है। तो वो चीज़ें पहले से ही तैयार रखी जाती है इन द previous phase, that is G1 phase. So it is actually a preparation for the next phase to commence. So this is how it is a coordinated process. It's a very important process. So यहाँ पे हम कहेंगे फिर now we understand that because the in S phase the DNA replication has to be done. For that purpose we need enzymes. We need enzymes, for example helicases, topoisomerases. We need DNA polymerases, so we need these enzymes which are required for DNA replication. These enzymes have to be already prepared. They are prepared in G1 phase. That means the protein synthesis takes place in G1 phase. That's why it's called the growth phase. It accumulates the nutrition. So these enzymes which are required in S phase are already prepared in G1 phase. We need nucleotides. हमें न्यूक्लियोटाइड्स की जरूरत पड़ेगी because you know that nucleotides are the building blocks of the nucleic acids. जो न्यूक्लियोटाइड्स होते हैं, nucleotides or the building blocks of nucleic acids. तो जो न्यूक्लियोटाइड्स होते हैं, न्यूक्लियोटाइड्स ये डीएनए न्यूक्लिक एसिड्स में आते हैं, डीएनए एंड आरएनए. वी हैव डीएनए एंड आरएनए, दीज आर द न्यूक्लिक एसिड्स. तो न्यूक्लिक एसिड जो है, these are formed from the monomers, basic units called the nucleotides. So unless there are not nucleotides present available, we are not in a position to form a new DNA, to form a new RNA. So these nucleotides have to be there in advance. So this preparation of the raw materials for the next stage, for the synthesis, for the replication of DNA ought to be produced in advance in the G1 phase. So these pooling of nucleotides, then formation of the enzymes, that's proteins, that means we have to form the proteins. Protein, because enzymes, all enzymes are proteins, we know that. जो enzymes होते हैं, they are proteins actually, except ribozyme. So these enzymes are proteins, so we have to form the protein. For that purpose, we have to pull in. We have to pull. The cell has to pull in amino acids, because proteins are formed from the building blocks, basic units, micromers, that's monomers, micromolecules. Those are called the amino acids. So pooling of amino acids, pooling of nucleotides, and then forming the, in the next step, the DNA, making of the enzymes, and duplication of the organelles, that take place in this G1 phase. That's, it's accumulating the nutrition, and then making all these things available for the S phase to commence. That means there must be protein synthesis, there must be enzyme formation, like the helicases, DNA polymerases, topoisomerases. There must be pooling of nucleotides for formation of, in the next step, that's DNA replication, adding the nucleotides and growing the chain. And all these prerequisites are done in the G1 phase. So it is the first phase, the first growth phase. That's when we talk about the time interval of these phases. We know that if we take a typical cell, it depends upon the cell, it varies from cell to cell and organism to organism. We can say, if we talk about the humans, if we talk about the human cell, let's talk about the human cell. A normal human cell divides, it takes about 24 hours. 24 hours to divide. It takes approximately 24 hours to divide and to complete the cell cycle. For this purpose, the 95% of the cell cycle time is consumed in the interphase. That means the most of the time which the cell takes, that's utilized in the interphase. So जो cell का बहुत ज़्यादा time होता है, that's utilized during the interphase. And in interphase, we can say most of the time, from this 91% duration of the time, it's also most of the time is utilized in the first G1 phase. It is the longest phase of the interphase. G1 phase is the longest phase of the interphase. We know interphase is the longest phase of the cell cycle, as it consumes 95% of the 95% of the cell cycle. If we take the example of a human cell, which is normally approximately dividing into 24 hours, 24 hours. That means we can say it will take approximately 11 hours in G1, 8 hours in S, 4 hours in G2, and 1 hour in M phase. So you can imagine here, you can now see 24-hour cycle. 24-hour cycle is utilization of the time, how the time is utilized in this interphase and in the whole cell cycle. 24-hour cell cycle, approximately 24-hour cell cycle taken by a human cell, a normal human cell in this cell cycle. So 24 hours duration, distribution of this time duration, 11 hours for the G1 phase, 8 hours for the S phase, 4 hours for the G2 phase, and 1 hour for the M phase. So that's why I wrote it here. It is about the 95% of the time duration in the cell cycle that's consumed in the interphase. You are now seeing here. So 95% of the time is consumed in the interphase. That's, if we count it, 11 plus 8 plus 4, it comes about 23 hours. Out of 24 hours, so we can see here, out of 24 hours, 23 hours are consumed in interphase. So you can now guess, it's a very important phase, interphase. And 23 hours, the most of the time of the cell cycle is consumed in the interphase, because it has to make the preparations. And when we see the phases of the interphase, For example, this is the G1 phase here. 11 hours. 11 hours for G1 phase. So most of the time of the interphase is consumed by the G1 phase. It's the growth phase. The cell has to accumulate the nutrition. Cell has to make the preparations for the next phase to commence. That's S phase. That's why it takes much time than the rest of the sub phases of the interphase, like S phase and G2 phase, then other phases. So this is it takes 11 hours, then 8 hours in S phase, 8 hours in S phase, and 4 hours in G2 phase. G2 phase, and 1 hour in total. We can say that it's only taking, it's only taking 1 hour. Approximately, it's not exactly, it's approximately takes 1 hour. That's 1 hour in M phase. So we can say here, most of the time which the cell utilize in the interphase, especially it is the G1 phase is the longest phase of the interphase. So all the preparations which has to be done, they are done in the G1 phase for the S phase to commence. That is, then after it's possible for the cell to replicate its DNA in the S phase, unless and until there are not all the requisites, all the availabilities which are required in the S phase, which are those things have to be made in the G1 phase. So it is the gap phase. It takes the time, it takes the gap from this previous mitosis to enter the next S phase, and it accumulates the nutrients. That's in the G1 phase, there is pooling of amino acids, pooling of nucleotides, formation of RNAs, and then duplication of the organelles, and then after formation of enzymes for the S phase to commence. Then after this G1 phase, preparations are made in the G1 phase. The cell now enters into the next phase, that's S phase. So this S phase is called the synthesis phase. The S phase is called synthesis phase. The cell will enter into S phase. This is called synthesis phase. Synthesis phase. So actually what this phase is, here the main important feature of the cell cycle takes place. The most important feature of the cell, we can say, as well as the interphase, that is the replication of DNA. Replication of DNA. This is the very vital step in the cell cycle. When the cell cycle from G1 phase enters into the S phase, it replicates, it doubles the amount of DNA. Its amount of DNA is doubled, because you know that from one cell we have to get the two cells. So to equally distribute the amount of DNA in the two daughter cells equally, the DNA has to be doubled, because each and every human cell, an organism, for example, human cell has 23 pairs of chromosomes. These have to be equally distributed. It's only possible when the DNA replication takes place and the amount of DNA is doubled. So this is a very, very vital stage, vital phase of the cell cycle, especially of the interphase. So replication of DNA and some histone proteins, formation of histone proteins. Histone proteins. तो histone proteins जो हम आगे पढ़ेंगे when we study about the the nucleotides, or we will study about the nucleus in detail, about the nucleosome concept. We will study it in detail, the nucleosome concept, what are histone proteins. But here we will say simply these are the proteins around which the long DNA wrap around. These chromosome, the long thread of chromosome 6' DNA, which is a very, very long DNA molecule. It's wrapped around the balls, like just like tense balls, wrapped around those proteins which are called histone proteins. These are found in the eukaryotes. So formation of the histone proteins are also taking place in the S phase to some extent. But the most important, the most vital step that's taking place in the S phase, that's the replication of the DNA, which is taking place in the S phase. That's why it's called synthesis phase. So when the cell enters into the S phase, it doubles its DNA. Now the cell is committed to divide, because double amount of DNA, it's not possible for the cell to encompass the double amount of DNA, because it could create abnormalities in the future cell. So when we can say, when the cell into S phase, and it quite nicely divides, replicates, doubles its amount of DNA, replicates its DNA, increase the doubles the amount of DNA, it is committed to divide. So we can say, हम कह सकते हैं कि जब सेल एस फेज में आ गई, इसने डीएनए को डुप्लीकेट किया, डीएनए का अमाउंट डबल किया, रेप्लिकेशन की, इसके बाद उसको डिवाइड होना पड़ता है, इट्स कमिटेड, उसको एक सिग्नल मिलती है कि आपको डिवाइड करना है. सो दिस in this S phase, the most important thing is that there is replication of DNA. So next phase is that is G2 phase, and we will deal in the next lecture, because we are continuing with this interphase. First we have started the G1 phase and then S phase. In the next phase we will talk about this phase and the later phase in the next video. So here we have studied about the first phase of the cell cycle, that's interphase, which is having three subphase: G1 phase, S phase, and G2 phase. G1 phase is the gap first phase. It is the time interval from the previous mitosis up to this synthetic phase, and it takes the time about 11 hours. In case of human cells, it's the longest phase of the interphase, and here the cell grows in size, and then after it moves from the cell moves from G1 phase to S phase. In S phase, there is the most important thing which happens, that's the replication of DNA. The amount of DNA is doubled, and there are also the formation of histone proteins. After that, the cell will enter into G2 phase. This phase and the next phase we are going to discuss in the later videos. Thank you for watching. Subscribe to our YouTube channel, like our videos, share our videos. For the next videos we will upload. Thanks for watching.